Hello, 7th grade. In our last lesson, we learned about cell theory and how cells were discovered when Mr. Hook looked under a microscope at some cork's bark, and he saw the cells in there. But today, we are going to dive a bit deeper than that and look inside a cell. So we're going to start to learn about how a cell actually works and what it looks like on the inside. And there's lots of really fascinating things here, so I hope that you will enjoy this lesson. So, to first understand how we know stuff about cells, let's look at the tools that we use to see cells, and that is the microscope. There are three types of microscopes that I want to tell you about, just briefly how they work. The light microscope, and then two types of electron microscopes, the scanning electron microscope and the transmission electron microscope. Understanding how these microscopes work will help us better understand how we can see into cells and learn stuff about them that way. And of course, a microscope is a very useful tool for studying cells because we can't really see anything about cells using just our naked eye. We have to use some tool to enhance our vision of the cells. The first type is a light microscope. This is the classic type of microscope that you'll think of. It's the kind that I have in my classroom and that you've probably used before. It works by shining light up through a sample, and then you look through a series of lenses to observe that sample. So the lenses all have some magnifying power, like a magnifying glass. And when you look through multiple lenses, it's like looking through multiple magnifying glasses, and it gets really powerful. So you can magnify things up to about 1,000 times larger than what they would normally appear, or so. So here you can see an image. I think this is of some sort of plant stem, I think. You can see different cells in here. Each of those little circles is a cell. And you can see there's different colors, and it looks like they have different jobs in there. And yeah, so this is a nice sample of a light microscope. What's happening is light is shining up through this thin slice of stem, and then you're seeing the cells on the inside then. But we have more powerful types of microscopes that can let us see even tinier than 1,000 times. We have some microscopes that can let us see or magnify things up to 1,000,000 times, which is quite tiny. And those are our electron microscopes. And they might not look exactly like what you think of when you think of a microscope. Down here in the corner I have a picture of an electron microscope. It looks like a bunch of computers hooked up to some fancy equipment, and that's basically what it is. So, like I said, there are two types of electron microscopes, the scanning and the transmission electron microscope, and we're going to talk about both of those here. The scanning electron microscope will produce these neat images that look like they're three-dimensional. Now this is actually a picture of different kinds of pollen grains, so that yellow dust that comes out of flowers. This is a picture of it through a scanning electron microscope. The way that it works is that you coat your sample in this layer of kind of a metal, and then you shoot electrons at it. And then you have your machine that kind of picks up the signals bounced back by the electrons and such. And it lets you see it that way. So you're not actually using light to look into it when you use electron microscope. You're using electrons to look at it. So a scanning electron microscope is cool because it gives you these three-dimensional images. But there's also the transmission electron microscope. The transmission electron microscope will give you more of what looks like a two-dimensional image. And the way it works, you very thinly slice a sample. So you might take a cell here, and you use technology to very thinly slice it, and then you can shoot electrons at it and get an image that way. So the scanning is kind of a three-dimensional image. It looks all 3D. The transmission looks flat in 2D. That's the difference between them. Now the disadvantage of using an electron microscope is that you can't study live creatures that way. Because if you're slicing a cell apart, it's not going to be living, or if you're coating it in metal, it's not going to be living. So you can only study dead cells this way. But still, it's a very powerful tool that lets us look inside those cells. So let's start to talk about kind of the anatomy of a cell, so the different parts that make it up. And that's what the rest of this lesson is going to be, is all these different parts of a cell. So just like when you look at any other living thing, like me, for example, I have skin and organs, like I have a stomach and a heart and veins and all that. I have all these different structures that help my body to work the way it's supposed to. Cells will also have lots of different structures that will help their bodies, or their cells, to work the way that they're supposed to. So the first three most basic pieces that you need to know are the cell membrane, the cytoplasm, and the nucleus. The cell membrane is the layer that goes on the outside of the cell. So in the picture I have here, it is the yellow line that's on the outside of that oval, which is the cell. It's kind of like our skin. It's the barrier between the outside of the cell and the inside of the cell. It holds everything in and keeps other stuff out. You can kind of think of cells as very, very tiny water balloons. And using that analogy, the cell membrane is the rubber balloony part of the balloon. Now, the membrane is actually kind of flexible, and you can let stuff through and let stuff out, and we'll talk about that in another lesson. There's ways to transport things in and out of the cell, which is pretty cool. But right now, you can just think of it as the barrier on the outside. Now, interestingly enough, there's also membranes that you can find inside the cell around different organelles, and we'll talk about that later. The second part you need to know is the cytoplasm. The cytoplasm is represented by what is white in this picture here. It is basically all of the fluid inside the cell. So using our water balloon analogy again, the balloon part itself is the membrane, and all the water inside the balloon is going to be the cytoplasm. It's just the stuff that is filling up the cell. Now, one little note on the word here, cyto often has to do with the word cells. So if you see a word that has cyto in it, think cell. So this is like cell plasm, or like cell plasma, sort of, something like that. Now, the next part that you should know is the nucleus. The nucleus, not all cells have a nucleus. We'll talk about which ones do and which ones don't later. But the nucleus contains all of the information for the cell. So it's kind of like the encyclopedia that holds all of the information that the cell needs to function. So it contains all of the DNA and the genetic material. So any kind of instructions that the cell needs to run can be found in the nucleus. And those instructions can get outside, and other parts can read them then. And we'll talk about that another time. Yeah, so these three basic parts right now are the membrane, which is the outside of the cell, the cytoplasm, which is the liquid inside the cell, and the nucleus, which is kind of, you can think of like the center of the cell that holds the genetic information. It holds the information of the cell. Now, the nucleus is actually floating around in the cytoplasm, and most of the stuff in the cell is just floating around in the cytoplasm, because the cytoplasm is just that liquid that everything's in. So the nucleus is found inside the cytoplasm. Okay, let's take a moment and talk about a few different kinds of cells. You know, I was talking about not all cells have nucleuses. The actual plural is nuclei. It's the plural of nucleus. Two main categories of cells you need to know are prokaryotic and eukaryotic cells. Eukaryotic cells have a nucleus, and they have other organelles. Now, like I said, the nucleus is that centerpiece that has all the information in it. And an organelle is basically any structure that is enclosed by a membrane. So kind of the same sort of material that you have on the outside of the cell, you can have that surrounding other structures inside the cell. So it looks like we have some, in this drawing here, some little pink or purple membrane enclosed things floating around. I don't know what they are. It's a very simple drawing. But you can think of these as organelles in here. And then we have our nucleus. So a eukaryotic cell has a nucleus, and it has other membrane-enclosed organelles. Now, a prokaryotic cell does not have a nucleus. I have a little diagram of one down here. Its genetic material, so all its information that it needs is just kind of floating out in its cytoplasm. It's just there. And you can see in the diagram there's kind of a squiggle right in the middle. That is the genetic information. That is its DNA just floating out in the middle of it. Now, a good way to kind of tell whether an animal or a creature cell is going to be eukaryotic or prokaryotic is to look at whether it's unicellular or multicellular. In general, this is just very general, most unicellular organisms are prokaryotic. So bacteria is a classic example of this. And this structure that's right here, this diagram, looks like a bacteria cell. It has a little tail to swim around and has its DNA just floating out in there. So lots of unicellular cells are going to be prokaryotic. And multicellular organisms are usually going to be eukaryotic. So you and I have eukaryotic cells, because our cells have all these fancy features inside, like organelles and nuclei and all that stuff. Also, fun fact, eukaryotic cells tend to be a lot bigger than prokaryotic. Prokaryotic, like bacteria, are very, very tiny compared to our cells. Okay, so what we're going to do now is we're going to talk about a bunch of these structures. And you're going to have to know all the structures that are found in a plant cell and all the structures that are found in an animal cell. Right now I have a picture of an animal cell up on the bottom right of the screen. And I'm going to be referring to this to teach you all of the structures that are found in both animal and plant cells. Basically, a plant cell has everything an animal cell has, but a few extra things, and I'll talk about those few extra things at the end. So I'm going to be going in a slightly different order than what your book is going in, but that's okay. I think it should still make sense for you. So the first structure we're going to talk about are ribosomes. Now, ribosomes are not actually enclosed in membranes, so not organelles in that sense, but they're still there and they're worth talking about. They translate information from the nucleus to build these important molecules called proteins. Now, proteins are the kind of like the main goal of your cells, to like build some sort of protein. Like this is something called the central dogma of biology, where basically it's all about cells building proteins. That's what a lot of life boils down to. It's very, very complicated, but also very simple in certain ways. Now, the nucleus, remember, has all that information that helps the cell to run smoothly. It has all the information for everything it needs to build and do and all that. And what it does is it actually sends out little bits of its code into the cytoplasm, and the ribosomes will grab a hold of those pieces of code, and they will translate them into proteins. So it's a really neat process that we'll talk about more some other time, but we're just giving a very brief overview right now. So ribosomes translate the information from the nucleus to build proteins. And by the way, all of these structures that I'm talking about, you will need to know them and you will need to have them memorized. They are important to know. So make sure you're taking good notes on all of this. So now we're going to be going through a series of different organelles, since the ribosome technically wasn't like an organelle, although it's usually counted in the same category because it's a structure found in the cell. The first one is mitochondria. The mitochondria is nicknamed the powerhouse of the cell, and it basically works as an energy generator. It is a power plant. It makes energy for the cell to use. Cells need to use energy, just like any living thing needs energy, and the mitochondria are what process food to make that energy. So they're these structures that they almost look like beans with little squiggles inside of them. And what they do is they take in sugar molecules and they can make it into a usable form of energy for the cell. And your cell is going to have multiple mitochondria, not just one. There's going to be a lot of them. So they are the powerhouses of the cell. The next organelle is going to be the endoplasmic reticulum, and that sounds nice and sciencey, doesn't it? It's this kind of foldy, envelopey looking thing that's right outside the nucleus. So our nucleus is this purplish color, this reddish purple color ball that's in the middle of this cell, and it sends out some information and to build proteins, and the ribosomes will start that, and the endoplasmic reticulum is also going to help process and build these proteins. So the endoplasmic reticulum is kind of like, imagine like a postal service or something like that. It processes these things and then it sends them places. So you can see in this diagram there's lots of folds here, and proteins are being built in these folds, and then it actually ends up pinching off and making these little bubbles called vesicles and sending those throughout the cell. So we'll go on to our next one, vesicles here. The vesicles are little packages that transport materials. They're basically like little bubbles that float around in the cell, and they can hold materials like maybe some unfinished proteins or finished proteins, and they'll move them from one place to another. And oftentimes, vesicles will be transporting proteins from the endoplasmic reticulum to this purple pancake-looking structure called the Golgi apparatus. The Golgi apparatus actually finishes processing protein. So this is all kind of like one big production line, like a factory almost. The ribosomes will do a lot of the work, and then sometimes the endoplasmic reticulum and the Golgi apparatus will also be part of the assembly line as well. Another type of bubble that you have floating around in the cell is something called a lysosome. A lysosome is nicknamed the stomach of the cell. And its job is the same as our stomach. It's to break down kind of materials to be used by the cell. So it could be parts of the cell that are no longer working that need to be broken down, like say some organelle stopped working and needs to be broken down, or maybe there's little pieces of excess protein floating around, or whatever. And basically it takes these materials and it'll break them down and recycle them so that the cell can use it in new ways. So it is the stomach of the cell. It takes things, breaks them down so they can be used, just like our stomachs. Okay, so those are all the parts that are in an animal cell that I want you to know for now. But there are a few parts that are special in plant cells that they have that we don't. And I want you guys to know those as well. So plant cells have all the things we just talked about and these other special things. The first is going to be the cell wall. So on the outside of all cells, you have a cell membrane, including plant cells. But plant cells also have this tough outer coating on their cell called the cell wall. And so that's going to be outside the cell membrane. So from outside to inside, you're going to have the cell wall, then the cell membrane, and then you have the cytoplasm in the inside of the cell. So this is this tough outer covering of the cell. It is outside the cell membrane. Plants have it, and we do not. They also have chloroplasts. So, you know that plants use sunlight to make their own food. They use it to make sugar in a process called photosynthesis. And the chloroplasts are the structures that actually do this job. So they have lots of different things inside the chloroplast, but one of those things is chlorophyll, which is that green pigment that makes plants green. And that's what's actually absorbing the sunlight and using that to make energy then. So you can see here we have these chloroplasts. They look kind of like the mitochondria, but rather than having a squiggle inside, they have like stacks of pancakes inside. Another thing that looks like stacks of pancakes. And this is a diagram of a plant cell now, by the way. It's not an animal cell anymore. The third part that I want you to know that is unique to plant cells is the central vacuole. Now, the central vacuole is really just this big, what looks like an empty space. So it sometimes takes up most of the cell. So you look here and you see this great big bubble-looking thing. That is a central vacuole. It is used to store water, waste, and other materials. That's one of its purposes. It's just kind of a big storage site for stuff that can be in fluid to go into. So cells can sometimes hold waste there before getting rid of it, or hold extra water there or extra materials there in this central vacuole. But additionally, it provides support for the plant. So, especially in smaller plants, there's this thing called turgor pressure, where you know how if you have a plant and you don't water it for a few days, it kind of starts to wilt and droop down, and then you water it and it springs back up? It actually has water inside the cells that are kind of like, it's pressurized. So imagine a balloon that is inflated and it's nice and firm because you have lots of air in that. And when you take that air away, it kind of gets saggy and it's not as strong of a balloon anymore. That's kind of the way the central vacuole helps plant cells stay supported. When it's nice and filled with water, it kind of puts this pressure on the cell that makes it nice and firm. But when it's low on water, it'll start to kind of sag and droop, and the whole plant will then follow that. All right, so I would like you in your notes to make a sketch of both a plant and an animal cell. I think this is going to be very helpful for you to understand these different structures. And I'd like you to label all of the structures that we talked about, so all of the organelles, in both the plant and animal cells, and then give a very brief description of what they are. I think this is going to be the most important piece of your notes, is having this nice diagram with that's labeled. So try and draw them in a way that looks like what we talked about the structures actually looking like, and sometimes they will look a little bit different depending on the diagram that you're referring to draw them. But you can find some nice drawings in your book on page 22, or you can also refer to the drawings that I had earlier in this slideshow. They should all be sufficient for drawing all the organelles and structures that you find inside a cell. So there's a list down below of the different things I would like you to draw: the cell membrane, cytoplasm, nucleus, cell wall, ribosome, mitochondria, chloroplast, endoplasmic reticulum, vesicle, Golgi apparatus, lysosome, and central vacuole. Remember, some of those are only in plant cells, and some of them are in both plant cells and animal cells. So make this nice drawing in your notes. Make it take some time. Don't rush through it. try and make it a good drawing. I think it'll be very useful for you to refer to when you're studying this stuff later. Additionally, I would like to sometime this week, and I'm thinking Thursday, do a virtual meeting with you guys. I think you guys have done a virtual meeting with one or two of your other classes, perhaps, using WebEx, and I would like to do that as well. Now, in the RenWeb homework notes, I'll put more information as far as the time and what we're going to be doing during that meeting. I'll let you know more details when the time actually comes for that. So you can be looking for that tomorrow or the next day. But let's aim for sometime Thursday morning, just so you can kind of have that in your sights. Anyways, I hope that you enjoyed this lesson on cells. And it's a lot to take in, I know, but the more time you spend looking at drawings and studying, it'll help you to learn all of these different structures. Remember, I would like you to have all of them memorized, like know all their names, how to spell them, and what they do for the cell. It'll be helpful going forward from here. Alright, well, I hope that you all have a great day. Take care.